All right, welcome everybody. Let's go ahead and get started. My name is Jen Tang, and I'm the Director for Federal and Community Relations at Lawrence Berkeley National Laboratory. And thank you so much for joining us this afternoon. Since 1931, which was our founding date, Berkeley Lab has been dedicated to advancing the scope of human knowledge and seeking science solutions to some of the greatest problems facing humankind. For those doing the math, this year is the lab's 90th birthday, and to mark the occasion, we've developed an array of content, including podcasts, virtual tours, historical highlights, a charitable giving campaign, this speaker series, and many more, all intended to shine a spotlight on the lab's unique approach to team-based and pioneering discovery science, which has resulted in broad societal impact. As we reflect on the past achievements and imagine what the next 90 years may have in store for us, we invite you to join us in our celebration. Please be sure to visit our 90th anniversary website to explore all of the different features and events. Now, before we get started, let me cover a few housekeeping items. I wanted to let folks know that we will be recording this webinar so people who aren't able to join can watch it later. You'll notice that your video and audio were automatically turned off when you joined the webinar, but don't worry, you'll be able to see the speakers and their presentations. And there will be a Q&A session following the speakers' presentations. What you can do is submit questions at any time during the presentations using the Q&A function at the bottom of the window. And before I introduce our first speaker, I just want to say a quick thank you to my colleagues in the Government and Community Relations Office, Strategic Communications, and IT. This event would not have happened without their help. So with that, I'm going to stop sharing my screen, and it is my pleasure to introduce Nigel Mouncey, the Director of the Department of Energy's Joint Genome Institute.
Well, good afternoon, everybody. Thank you for joining us today. And it's my pleasure to sort of introduce the session today. And you're going to hear about how Berkeley Lab researchers are leveraging our multidisciplinary expertise and our 5 national scientific user facilities at Berkeley Lab that are accessible to collaborators worldwide, and how we're using these capabilities to develop high-impact solutions to the COVID-19 crisis. So Berkeley Lab has a history of supporting the nation during national emergencies. And the 3 examples on this slide are the kinds of emergencies the lab has played a role in to date. With Deepwater Horizon, the lab leveraged its expertise in modeling and simulation to help assess the environmental impact of the spill, including understanding mechanisms for oil degradation from all of the principal oil degrading bacteria that were observed in the original oil spill, and also helped to develop ways to cap the well. With Aliso Canyon, which is a very large national gas storage facility, the lab used software developed at the lab to simulate the behavior of the leaking well, and showed that a relief well would be effective in stopping the leak, which was actually how what was employed to indeed stop the leak. And with Ebola, pharmaceutical researchers utilized one of our user facilities, the Advanced Light Source, to help map the structure of the virus, which helped lead to the development of the Ebola vaccine. So the lab was really primed to respond when the COVID-19 pandemic hit us all last year. And the Department of Energy created a coordinated response across all of the 17 national labs under the National Virtual Biotechnology Lab, or NVBL. And Berkeley Lab also participated in the National COVID-19 High Performance Computing Consortium, which also included industry members. Our researchers have focused their efforts on better understanding and combating the virus and disease. So COVID-19 is caused by the SARS-CoV-2 virus, a member of the coronavirus family. And this virus has proteins on its outside that enable it to stick to human cells. The virus then enters human cells and hijacks the human cell machinery to make many more copies of itself. And these proteins on the outside of the virus that allow the virus to stick are the basis of the vaccines that are currently being distributed that stimulate the body to make antibodies that block these proteins and prevent the virus from infecting human cells. Other areas of research focus on therapeutic treatments that prevent the virus from making these proteins, or proteins that block the interaction of these proteins with human cells. And another important area is detecting the virus and investigating how the virus spreads. And so, like all of us, Berkeley Lab has had to adapt to the restrictions that the COVID-19 pandemic's placed on our lives, but we continued our operations to enable the essential COVID-19 R&D that you'll hear about today. We started many internal projects. We've worked with partners in industry and at UC Berkeley. And we're very grateful to have received $12 million of funding from the CARES Act that's largely gone to funding multi-lab projects under the MVBL and also to purchase new state-of-the-science equipment to support our COVID-19 R&D work. We've also rescoped current funding towards computational projects from the Exascale computing project. And we've also looked to the future to build a vision and a strategy for COVID-based research that can establish or expand capabilities across scientific areas for future DOE mission research. And this is a lab-wide vision that identifies strategies for new platform technologies and new research. And some of the capabilities proposed are specific to addressing COVID-19, but many more would really lay the groundwork for longer-term capabilities that could be applied to other research topics as well. And this strategy has 2- and 10-year goals, along with metrics and milestones that allow us to assess progress. And this plan was put together from a series of workshops that we held at the lab that included around 40 of our scientists at the lab. But of course, we've had many more of our incredible Berkeley Lab team work together on COVID-19 research. More than 275 of our science and operations staff have been involved in conducting and supporting the COVID-19 research over the last year. And you're going to hear from just a few of those very shortly. So Berkeley Lab has an amazing array of state-of-the-art user and other unique facilities. In fact, we have more user facilities and a wider array than any other national lab. And all of them have had an important and a varied role to play in research to address COVID-19. So one of these user facilities is the Joint Genome Institute, and I have the privilege and pleasure of leading that institute. And normally we focus our efforts on serving the global user community with large scale and advanced genomics science capabilities, such as DNA sequencing, that support the Department of Energy's mission in energy and the environment. We have extensive expertise in viral genomics and in high throughput automation and molecular biology. And really over the last year, we've used our expertise in several different ways for COVID-19 research. Firstly, we established a new dedicated lab for developing and optimizing high throughput diagnostic assays, including pooling strategies that really increase the number of assays that can be conducted. And we deployed these methods at the Innovative Genomics Institute that's led by Jennifer Doudna, one of last year's Nobel Prize winners, at UC Berkeley to support their diagnostic testing of UC Berkeley staff, students, and for the local community. Secondly, we are also working building an understanding of how virus-host interactions occur, what the drivers are for those interactions, and doing this using model virus host systems. Thirdly, we've also been exploring the evolution of one of the proteins of the SARS-CoV-2 virus that plays a role in how the virus evades the immune system. And lastly, we've also performed extensive analysis of SARS-CoV-2 genomes to identify regions of the genome that seem to be more invariable and that can serve as better sites for diagnostic tests. Well, I think you're all probably aware now that the virus is mutating, it's making genetic changes, and we've seen a number of what we call variants, different versions of the virus that are emerging and spreading around the world that may have increased transmissibility and or may cause more severe disease. And so if we're better able to comprehensively track and predict the emergence and spread of these variants, this will help us better manage prevention and treatment of COVID-19. So we've developed a concept for viral variant surveillance to bring different data types, not only from clinical samples, but those from, let's say, wastewater, air filters, surfaces, together, and bring these data together for analysis and modeling that can be used to drive approaches for diagnostics, therapeutics, and vaccines. And we think this idea, this concept, really augments current efforts on variants that are taking place around the world. So I hope with this little introduction here that this has provided an overview of how Berkeley Lab has used our expertise and capabilities in the fight against COVID-19 to keep the nation safe. And you'll now hear more about the exciting work that our scientists at the lab are doing over the last year. And so it's my pleasure now to hand over to Corie Ralston, who's the director of the Biological Nanostructures facility at the Molecular Foundry.
All right, well, good afternoon, everybody. Thank you for having me speak today. So I'm very happy to tell you today about some of the protein structural work that we've done at Berkeley Lab and how this has really helped us hopefully bring an end soon to this pandemic. So the first thing I want to do is I want to give you a sense of scale, a sense of the size of proteins. So this is 1 millimeter scale, and of course our human eyes can see on this scale, but let's reduce by 1 order of magnitude and go down to 1/10 of a millimeter. So this is about the size of a human hair, and of course we can see human hairs, but we really can't see the level of detail that you can see in this microscope image of a human hair. Let's go down another order of magnitude now. Now we're at the 100th of a millimeter. This is a human red blood cells, but we can no longer see at this level. So we need microscopes now to be able to see, and to see things at this level. And we've only gone down 2 orders of magnitude. We'll go down 1 more now, and we're at a level of a micron. A lot of bacteria are about this size, and this is about the size where visible light microscopes start not to work anymore, and we need other types of microscopes. So at this point, we often turn to electrons to image things at this scale. So here right now this is an image of our current most popular virus, the SARS-CoV-2 virus. They're about 100 nanometers across, and you can see this image is really quite good. This is an electron micrograph image of the virus. But what you can't see very well is the proteins on the surface of this virus. So you have to go down another order of magnitude now to about 10 nanometers, and now we're at the protein level. So now you can start to see the level of detail that we really need in order to design drugs to fight this virus. So this spike protein that sits on the surface of the coronavirus is actually very important to the coronavirus, and it's important to us because it is what allows the coronavirus to recognize and to invade human cells. Now we have to actually go down 1 more order of magnitude to about a nanometer in order to get really fine structural details. So, and we need those details in order to design drugs. We need to know, for instance, that this carbon atom is exactly 0.2 nanometers from this oxygen atom. So we've gone now 6 orders of magnitude. And I wanted to remind you that at 2 orders of magnitude, we can no longer trust our human eyes to see anything. And at 3, we can no longer use visible light microscopes. So at this level of detail, we start to turn to even more sophisticated methods. So often we use a method called protein crystallography, which uses X-rays to get images at this level. And fortunately, Berkeley Lab has a lot of the instrumentation and the methods to be able to get images like this. So we have the advanced light source, which produces X-rays of exactly the right wavelength so that we can use this method called crystallography to get structural details at the atomic level. So let's go back to the spike protein for a second. So this is the protein that sits on the coronavirus surface, and it's used to invade human cells. So how do we stop this? How do we stop the coronavirus? So one way is to design antibodies that will recognize and bind the spike protein and help our immune system recognize it. So you'll see that an antibody is about the same size as a spike protein. So in order to get the details of the interaction between antibodies and the spike protein, we need to use protein crystallography to get the exact interaction region. And a number of researchers came to Berkeley Lab over the past year, and they solved antibody structures that were bound to the spike protein. And I'm very happy to say that they found some very good acting antibodies, and there are now a number of them in clinical trials. So now I want to tell you about another structural method that we've been using. So first I'll tell you about nanobodies. So nanobodies are about 1/10 of the size of an antibody. Some animals produce nanobodies, humans don't, but some animals do produce nanobodies in addition to antibodies. And it used to be up till a couple of years ago, we had to, in order to get nanobodies, we had to immunize animals and then purify the nanobodies out of their blood. But this group at UCSF very recently discovered a way to synthesize millions of synthetic nanobodies using yeast cells. So they used this method to create a couple billion different synthetic nanobodies that bound to the spike protein. And they narrowed them down to about a dozen that bound really well. And each of these bound at different positions on the spike protein. They bound with different strengths, with different levels of effectiveness. And then what they did is they bound some of these nanobodies together. And the reason they did this is because proteins don't sit still. So this is a simulation of the spike protein on the coronavirus surface. You can see that they're in motion. And in order to be effective, the spike protein actually has to change its configuration a little bit. So what this group did is they bound these 3 nanobodies together so that they effectively lock the spike protein into position, and the spike can no longer move, so it can no longer invade a human cell. And these nanobodies are so stable that they then turn them into an aerosol spray. And if you want to read about this, there's a really nice story here at this link. This is what they're calling AeroNabs. This was based on work done in just the last couple of months at Berkeley Lab. And they've turned this product into a commercial product. They're about to start clinical trials soon. So that gives you a very, very brief overview of some of the structural work that we've done at Berkeley Lab. I hope it gives you a sense of what we're doing and how we're helping fight to end this pandemic. Now I will stop share. And it is my great pleasure to now introduce Thomas Schenkel, who is the Interim Director of the Accelerator Technology and Applied Physics Division at Berkeley Lab.
Yes, thank you, Corie. I appreciate that introduction, and I'm just so amazed what you can do with these precision techniques of mass spectrometry and using the tools we have at Berkeley Lab. So I want to tell you a little bit about our work here in the Accelerator Technology and Applied Physics Division in the physical sciences area at Berkeley Lab in collaboration with Antoine Snijders from Biological Science and Engineering Division. You see here in the background an image of the laser bay of the Bella petawatt laser, one of the most powerful lasers in the world right now. And we make the connection to COVID pandemic research and prevention using this high power laser. So when we look at our the history at the laboratory, and of course started with Lawrence, Ernest London Lawrence, who invented the cyclotron. What's amazing about that machine at the time was that it completely opened the whole, it kicked the door wide open, it opened a whole new dimension of study, looking into the atoms and into the nucleus, things that people had not been able to do, and has had tremendous impact. It created a whole field, for example, of nuclear medicine and exploration in nuclear physics and particle physics that's continuing today. Lawrence's spirit of changing what's possible with tools for science, discovery science, is very much alive. And so here we have, instead of using microwaves to drive a cyclotron, we now have powerful lasers that drive the particle acceleration in a much more compact way, as shown here in these images from the Bella Center. And then together with that, we also develop high power magnets, and we put these beams to work at Berkeley Lab, also supported by powerful simulations, as shown here with the particle in cell code that track what's going on when these intense laser pulses impinge on matter, form a plasma, and then accelerate electrons and also ions. So when the pandemic hit back in, it seems like a long time ago, everybody was asked themselves, how can we help? And the best answer for many people might be just stay out of the way. But as scientists, of course, we like to think that look at ways how we can pivot our tools and apply our tools to urgent questions. And what we then soon realized as the research on COVID accelerated very quickly, that transmission is through droplets and aerosols, and you heard a little bit about that, and you'll hear more about that later. So in our context here, so we asked the question if we can study the virus inside droplets to learn more about its behavior and how long does it live, what sticks to it, what's next to it, what can we do about it? How can we do fundamental science to support understanding of the virus and then contribute to the prevention of the shortening of this pandemic and mitigation of future pandemics. So you already saw from Corie this image of the coronavirus with the spike protein, and Nigel talked about the spike protein. So we have here one way to look at it is with X-ray scattering, so you get amazingly precise atomic precision structure. But when I look at a droplet, I'm asking the question: how can we take this droplet where you have like a few viruses in a 10-micron droplet. So it's like a needle in a haystack. And how can we be clever about interrogating that droplet and learn to kind of dissect it and learn about the virus in it. So this may look a little complicated, but we combine here a series of laser pulses that drives and particle pulses and X-rays to shine a pulse of radiation on the droplet that can image the droplet and look at where the viruses sit. Does it sit, for example, at the middle or at the edges? And then we can fire a second and a third and a fourth laser into that cloud and dissect it. And in the limit, we can detect then, we will be able to detect single viruses and its fragments and kind of reconstruct the microenvironment of the virus in droplets. That's the vision. And so we started this kind of on a dime early on in last spring, basically. And an example here is shown in the demonstration that we had, that we pulled off, is that we can see the spike protein, so that's the culprit. And now it's not atomic resolution, but we can see the signature of that spike protein in a technique that's called mass spectrometry, where we excite and ionize and then analyze the result, the constituents of a droplet. And we found ways to enhance that signal. We're very excited about that. And then in the next steps, we like to take this from in two directions. On the one direction, we want to continue this research and apply our precision control of lasers and particle pulses to the precise imaging of droplets combined with mass spectrometry to analyze the constituents and dissect the virus and what's around it, characterize its microenvironment. In parallel, we also want to take it into a compact instrument. And right now we're doing this at the Bella Center with our precision pulses and the repetition rate of these pulses about 1 per second. We have an initiative proposed to the Department of Energy that we're very excited about. It's called K-Bella, where we will increase the repetition rate from a few hertz to 1,000 hertz, 1,000 times a second to do much more rapid imaging, and then develop in parallel also develop based on our knowledge that we gain a portable instrument that could be fielded like at airports or in hospitals to scan the air for the presence of virus. So this is what I wanted to share with you today about our work at Berkeley Lab at the Accelerator Technology and Applied Physics Division as part of team science, as part of a context of excellence and expertise in many areas. So with that, let me introduce our next speaker, Jeff Urban, who is the director of the Inorganic Nanostructure facility at the Molecular Foundry.
Yeah, I'm excited for the opportunity today to tell you about some of the work that we've been doing here in Berkeley Labs. In my labs, part of a collaboration in order to address some of the aspects of the pandemic. And I'm going to be telling you about work that we're doing on rechargeable custom fit PPE antiviral masks. So just for context, I think this has been laid out pretty clearly, but the extent of this pandemic has just been totally catastrophic and difficult to overstate. And as with many problems like this, there are really a long list of needs that accompany that in order to ameliorate and help society move forward. One that we focused early on and drew our attention was really the need for better masks. And this was, highlighted by many media outlets and many scientific outlets, and the need continues actually. And, this is as recent as last November, December, where there was a surge of the virus, at least locally, California and the Bay Area and throughout the state. And, really a lot of facilities, care facilities and other places were really running quite short on PPE. Personally, even within my family, I have 3 or 4 doctors in the extended family. And yeah, it was very concerning about, shortages in PPE and ways that they were asked to consider, reusing them or cleansing them. And it made it a very, it hit home very closely for me to see if we could do something to contribute and to help out. So the problem with a lot of masks, there are a few problems that we thought we could help, invest some energy and improve. One of them is really just a kind of a supply chain issue. You know, there's such a massive problem really getting products made that, you know, the notion that you just use a mask once and throw it away, you know, if you're working in a hospital or if you're a frontline worker, that really runs, you know, you run out of the capacity to do that. And, you know, the question becomes, you know, why are they kind of single use in most cases? And it's really kind of for a very simple reason. They're hard to clean. You know, you have to treat every time that you're using a mask in a situation as though it's contaminated, even if you maybe don't know. And even within that, they have a limited lifetime. And, you know, you, again, don't want to deal with them afterwards as a potential contamination risk. So our overarching goal, I mean, the thing that we thought we could contribute was to develop a rechargeable mask that had a custom fit, because we know if masks don't fit properly, that there are other, you know, they're not really as secure as they could be, and there are other avenues for viral invasion. So that was the principle that we were trying to explore. And, you know, the concept behind a rechargeable mask is actually fairly simple. So a standard kind of 3-ply mask or an N95 mask looks like this cartoon in the upper left-hand corner. And, you know, really the principle driving it, or the principle of action for the most part, is just electrostatic. You know, there are charges that come implanted on this filtration layer that actually immobilize viruses and, you know, for the most part, deactivate them. But this tends to fade over time, even within a day, depending on usage and conditions and humidity of all that. And, you know, again, it can't be restored. So even if you want to do UV, ozone, something like that, the actual, you know, sort of operative mechanism of the mask becomes degraded over time. So our concept was, you know, to take essentially that central principle of action, you know, that electrostatic mechanism, and incorporate it into a custom fit mask such that it could be charged. You know, you would use it for a day, maybe it would lose its charge, and then you could reinstate that charge, you know, using a kind of a test charging station that we had made. Okay, and it would have all the other, you know, necessary features of a mask, and also incorporate the important principle of, you know, correct fitting. The mask that we're making here and we've made is, you know, kind of premised on 3D printing, which, you know, many of you have probably seen or familiar with. So it gives you the ability to really kind of take custom diagnostics of face shape, or even if you grow a beard or your face changes or do something, and then get masks that are really custom built for that principle. And here's just a little graphic of what it looks like. Again, this is kind of uniquely enabled by a lot of the tools at the lab. You know, some of the graphics you're seeing here that look cool are taken using electron microscopes here that really allow us to take a look at the molecular structure in a fine way of the mask and see immobilized particles. So just to give you some insight into our process, we also had to build a particle filtration stand at the lab to kind of help us test how well the masks were performing. Prior to the pandemic, we didn't have, you know, kind of an aerosol testing station set up in the lab. So this was, you know, another important capability we've added to the lab network now, and something that we'd have ready for future use. And the idea here is that the rather simple looking nebulizer over there makes aerosolized mixtures that kind of closely mimic what you would get from a cough, or, you know, what you would get from some other kind of exudate. And then we pass this through our filtration device, and we test how efficacious it is at kind of filtering those particles as a function of size. And we measure that on the back end with a particle counter. And then what we're able to do is, over time, we recharge that mask using the power supply there, and then make the measurements again. And this just gives you a graphic on what that looks like in practice. This is just the really fine mesh filter material, and we're recharging it just, you know, using a very basic set of electrodes. And in practice, this would come, you know, kind of encased in a charging station that you see in the lower right. And, you know, again, this is just a different crosscut of a mask with the different filters and kind of state of charge. And, you know, I want to point out that, you know, this isn't necessarily directly translated to an N95 mask. There are different standards used here, but, you know, we can get into those details if people are interested in that, but it upholds the filtration principles quite well. And this is a graphic of the, you know, kind of as out of the box mask and the electrostatic charge in each of those layers in blue. And in red is the amount of charge that we're able to kind of reinvigorate the mask with. And this translates directly to performance. So the y-axis in this new graph is kind of filtration efficiency as a function of particle size. And you'll see that the mask fresh out of the box, we're able to, basically recapture quite a good fraction of that performance with our recharged masks. So moving forward, we're continuing to kind of, put the final touches on this product. And we've had a lot of interest from manufacturers and companies and partnering with health providers as well. So we find that, very exciting because we hear what the needs, truly are, and we're able to get closer to addressing that. We also have a kind of second generation we're working on with unique materials that we've made in order to make it self-cleansing. And again, living in California, this isn't just for kind of pandemic and biological threats, but would also work very well whenever there's particulate matter in the air. So if there's a fire nearby or something like that as well. And again, I want to highlight the team aspect of what's going on here. This has been a very collaborative team and a very strong team. Peter Hoseman has played an absolutely giant role in this as well. And the postdocs and students here listed have, really driven the project with, just tireless dedication. So I'm going to stop my screen now, and I'm going to introduce Tom Kirchstetter, who's the director for the Energy Analysis and Environmental Impacts Division. And I think, Tom, you can take it away.
Well, I just want to say, I feel like I'm learning so much from my colleagues. I'm really enjoying this event myself. So my name is Tom Kirchstetter, and I study air pollution, particularly emission sources and emission control technologies. And typically I consider emission sources like cars and trucks. But last year, my colleagues and I, along with the other research that you're hearing, we pivoted to do research. And in this case, we wanted to better understand the transport of SARS-CoV-2 emitted from people in indoor spaces. And that's what I'm going to tell you about in this presentation. So SARS-CoV-2 is emitted inside of respiratory fluid particles, as illustrated on this slide. And those fluid particles range in size from 5 to, say, 1,000 times larger than the size of a single virion, which is the way we call a single entity of virus. And the large particles, as you heard, they're often referred to as droplets, and those are large enough and settle quickly to the floor on the order of seconds. The smaller particles are often referred to as aerosols, and those settle much slower and can remain airborne for minutes to hours. So the larger droplets that settle quickly can expose somebody who's standing close by, as shown in this figure. But the smaller respiratory aerosols that stay suspended for minutes to hours can travel well beyond 6 feet, so that both nearby and farther away individuals can be exposed to these smaller respiratory aerosols that may contain infectious SARS-CoV-2. Now, since these aerosols disperse throughout the room, as sort of illustrated by the dots all over the room here, the building's heating, ventilation, and air conditioning system, which I'll refer to as the HVAC system, that can actually reduce the concentration in the room by replacing polluted air with clean air. And in that way, the building's or the room's HVAC system may actually lower transmission risk. And that's what we're looking into in this research. And to do that, we leveraged one of the many unique resources at Berkeley Lab. This is a picture of the Flex Lab, which is a one-of-a-kind facility designed to test the performance of building energy efficiency technologies. Now, for this research, we used it to study how overhead heating, ventilation, and air conditioning systems affect the transport of aerosols in meeting rooms and classrooms. Here, I show you a chamber in the Flex Lab that we've configured as a socially distanced conference room, where we have heated mannequins used in place of humans, and where the room has been outfitted with 100 environmental measurements of air temperature and velocity, which help us understand air mixing in the room, and the concentrations of gases and airborne particles are measured to help us understand exposures. And the poles that you see in the photo are used to suspend many of these measurements at various heights in the room between the floor and the ceiling. Here you see the chamber configured as a classroom. And in this particular experiment, we had placed a physical barrier in front of the instructor. And you can also see elements of the overhead HVAC system. For example, in the rear of the room by the lights on the ceiling, you see 2 diffusers that supply air to the room. And then here in the front of the room, in the structure, is a return. This is the exhaust of the room. This is where air is returned back into the HVAC system. And if these look familiar to you, it's because overhead HVAC systems are the most common way that conditioned air is applied to and removed from meeting rooms and in many classrooms. Now, fortunately, to study how virus-laden respiratory fluid aerosols are transported in indoor spaces, we don't actually have to work with any virus. Instead, we emitted carbon dioxide gas, which is going to disperse in a room much like very small aerosols. And we safely generated aerosol particles, including larger particles that behave differently. And these were emitted in a manner that mimicked the air velocity from the mouth of a seated person, as shown here, right next to this mannequin's head. Just for illustration purposes, I'd also point out that in the top right, these are some pictures of many of our optical particle counters. These count the concentration of particles in the room. And in the center, we have a mannequin very hard at work, wearing one of our optical particle counters. Now, in some of our experiments, the HVAC system is providing air that was warmer than the room, i.e. heating the room. And in others, it was supplying air at the same temperature of the room, which we call neutral. And I'm showing you some data from these 2 sorts of experiments. This plot shows the measured temperatures in the room at various heights. In the room with a 9-foot ceiling, as illustrated here, 9 feet is about 2.75 meters on this scale. And when the supply air was neutral in temperature, what we see is that the room air temperature is the same from the bottom to the top. But when the supply air was warmer than the room, a temperature gradient is formed, shown in the red lines, where the warmer air that's being supplied by the HVAC system near the ceiling actually stays on top of the colder air or the cooler air down below. And this is significant because it inhibits the mixing of clean air supplied to the room. And the implications of that should become clearer here. This plot shows the results of one of our experiments where we're measuring exposure concentrations around the room, both for the case of the neutral air supply and the warm air supply. And we measure the concentrations at 3 different heights, at 1 foot above the floor, near the ceiling, just 1 foot below the ceiling, and then at the mannequin's mouth, which is 3.5 feet above the floor when the mannequin is seated. And we're going to call this the breathing zone. And you can see the results differ between the neutral air and the warm air supply. So due to the poor mixing when the room is heated from the ceiling, the HVAC system that supplies air to the top of the room, well, that air remains near the ceiling. And then the HVAC system just removes that same air. So you can see that as a result, the concentration is actually lowest near the ceiling. Now, since the supply air doesn't effectively mix down into the whole room, anything expelled in the breathing zone just accumulates in the breathing zone, which can lead to much higher concentrations at locations throughout the room. And this is what we call a Boxen-Riskos plot. It's one way to show the distribution of measured concentrations throughout the experiment. And so most of the data here, you can see you can have concentrations, say 4, 5, 6 times higher than you would if the room was well mixed. So in other words, in order for the HVAC system to provide the benefit of lowering exposure concentrations where people are breathing, it's important that clean air supply mixes down into the room. Now, we found that adding 2 small portable air cleaners provided enough air movement to overcome this limitation. So these units, one of which you could see here in the photo next to the mannequin, they draw air into the front and then shoot it up towards the ceiling, which induces mixing. And I've added to the plot the results when the air cleaners were on, and you could see that the concentrations are again then much lower and typical of what would be expected for a well-mixed room, where the HVAC system is effectively replacing polluted air with clean air throughout the entire volume of the room, rather than just near the ceiling. So in short, air cleaners are a good way to help ensure mixing in rooms. And they would also filter the air, which provides an additional safety measure by removing airborne particles, which is something that Jeff just referred to in the masks. So we're in the process of formalizing these results for submission to a peer-reviewed journal. And that's an important part of our scientific process because the results will undergo careful inspection by experts in the field. And then if it passes that review, it's going to be published. And then the lab's communication team, led by John German, whose contact I have here on the slide, helps us disseminate the information to the public. And lastly, I'd just like to say that these are all the researchers who pivoted to work almost exclusively on this project for the past 6 months. And so with that, I'd like to stop sharing and turn the mic over to Amalie Trewartha, a postdoc in our material science division,
Okay, so I'm going to be talking about something a little bit different from the others. I'm talking about the Covert Scholar project, which is a project that started here at the lab with kind of the overarching goal of trying to use AI to help scientists get to grips with the enormous amount of COVID-19 research that's been coming out and to sort of get the right information in the right hands as quickly as possible. For a little bit of my background, so I'm a materials scientist as my day job, and I work on the MathScholar project. So one of the biggest problems in material science and in science more generally is that the vast majority of scientific information is only available in the form of sort of unstructured text in scientific papers. And so the only way to access that information is to like sit down and physically read the paper. But the scientific literature is getting so big and so many papers are coming out, it's just utterly impossible to read every paper that comes out. There's more papers than any human can read in a lifetime every year. So the MathScholar project started with the idea of trying to use tools from AI in natural language processing to help extract that information from text and make it available to scientists in a sort of usable form. Doing things like making a database of experiments that have previously been done. So if you're thinking of doing an experiment, you can check, see if it's been done, see if something previous has been done, things like that. And so on the back of that, relatively early on in the pandemic, we were contacted by some of our colleagues in the Innovative Genomics Institute here at the lab, who were having trouble staying on top of the enormous amount of COVID-19 research that was coming out, which at the time they were tracking with a Google Doc. So this is a plot I made of the number of COVID publications that came out in the first 10 months of 2020. COVID had this enormous rapid research response and research emerged very quickly. Like you can see even by as early as April, which is like soon after we went into lockdown, there were literally tens of thousands of COVID publications out and papers are coming out at the rate of hundreds a day. So even if you're looking at a relatively small subfield of that, it's hard to just look at all of those papers, let alone read them. And it wasn't just about the number of papers coming out, the ways in which those papers were coming out changed. So under normal circumstances, scientific papers go through what's called peer review. So when you submit a manuscript to a journal, the editor will send it out to some subject area experts who will check it over for validity of the methods and the results and how important it is. Sort of a quality control, which lets you be assured when you read a paper in a journal that the results are essentially correct. But this process takes months or even up to a year, which in the age of COVID, when results are needed on the order of weeks to inform public policy, is just too slow. So scientists increasingly turn to what's called preprint servers, which is a way that you can put your paper online as soon as you've written it and make it available to other scientists while you go through the peer review process. And so this has the advantage that people can read your research much sooner, but it does mean that this quality control that journals provided is gone. And it also means that the kind of places in which the papers appear are different. So you might be used to looking at journals to find the most important information in your field. And then all of a sudden at the start of COVID, the most important research was appearing on these scattered preprint servers. And so people didn't necessarily know where to look to find it. Just to make a point about how big and new this shift is, this is a New York Times headline I found from 2016 that describes preprint servers as going rogue and publishing directly to the internet, which is a little bit extra, but it does kind of make the point that this is like a very new and very big shift. And I think it's something that will persist after COVID as a genuine shift in the way people publish papers in these fields. So to kind of help people deal with this, we created the Covert Scholar project, which we designed as a sort of one-stop shop for COVID research. So we collect all COVID publications as they come out across all of these different preprint servers and all of these different open access sources and make them searchable on our website. And we use AI tools to do things like tag them with what the subject area is or suggest some related research or what the keywords are. So if you have a question like, what are the papers on epidemiology that came out in the past week? That can be answered in a couple of clicks on our website, and it's not really doable on the existing scientific literature search tools. And thanks to an enormous amount of support from our colleagues at NERSC, the computing center at the lab, we were able to get this up and running very quickly. This was up and running by April last year. It was a matter of weeks from when we started that we had this up. And at this point, we have over 120,000 different COVID research publications and another 70,000 on related topics. So things like SARS and MERS and similar viruses. And we collect these from 19 different preprint and open access sources. We've had pretty good uptake among the scientific community. We have over 5,000 monthly users worldwide, which is a pretty significant number of scientists. The scientific community is relatively small. And so it's not just about getting individual papers in people's hands. There are insights you can get from having a large amount of text in one place that you cannot get from reading papers in isolation. And so I thought I would do a quick demo of a tool on our website. So this is a word embedding projector. And so each of these dots represents a word that's used in our COVID research collection. And how close it is to the other dots represents how similarly those two words are used in the collection. So as an example, like this, I've taken spike protein, an important coronavirus protein, and I found the 100 nearest neighbors of it. And so, I mean, the closest neighbors tend to be things like synonyms like S protein. But you can also see here ACE2, which is the human receptor that the spike protein binds to, is listed as a very similar word. And so there's some kind of genuine scientific information available here. And while this might be an example that's kind of well known among scientists, we've been using it to do some more sort of exploratory analysis, like finding candidates for drug repurposing that maybe we see are used very similar to proteins that turn out to be important for coronavirus. We have also been working with the open access journal Rapid Reviews COVID-19 to try to accelerate the peer review process so we can get peer-reviewed papers out there faster. So we've been building AI tools that let editors work hand in hand with the AI to get the papers to the right reviewers quickly. And partially thanks to our tools, they've been able to get the peer review timeline down to 2 to 3 weeks from a paper appearing from months and months. Yeah, so I would like to thank the Covert Scholar team who did an enormous amount of work and had very many late nights when we were getting this up and running, especially John Dagdal and Hao Yan Huo. And NVBL, the lab's LDRD program, and C3.ai, Digital Transformation Institute, for funding us. Now I believe we're turning over to Jen and Dan for questions.
Thanks so much, Amalie, for that presentation. And to all of our presenters today, I'm actually going to ask everybody to join us back on screen. Turn your cameras on and your microphones on. And I'm also going to ask my colleague, Dan Krotz from our Strategic Communications Office to join us for the Q&A session. So as a reminder, people can actually submit questions using the chat function at the bottom of the Zoom window. And I wanted to start first with a question that we received from our audience. And this one is for you, Corie. Could you explain to us what the benefit is of X-ray crystallography over cryo-TEM? And for those in the audience who aren't technical experts, could you start with an explanation of what cryo-TEM is?
Sure, yeah. So cryo-EM, which is the more general term, is a cryoelectron microscopy. And it's a method that uses electrons to get images. It's essentially, there's a number of ways to do it, but you can essentially shoot electrons at, say, virus particles on a grid, and then look at the sort of the shadow that it creates, and from that figure out what the shape and the structure of that particle is. So that's distinct from X-ray crystallography, which uses X-rays and basically shoots X-rays at a sample and measures how they are diffracted off of that sample, and uses that information to reconstruct the structure of the protein that you have been shooting at. So the first thing I want to say is that, these days in science, it's pretty rare that you only use one method when you're trying to solve a problem. So people generally don't just use crystallography and don't just use cryo-EM. They use a lot of different methods to get at the information they're trying to get at. And I wouldn't say that any one method is necessarily better than another. They all have strengths and weaknesses. Very generally, the crystallography can see fine atomic detail that usually you can't see using cryo-EM. So you can actually look at a drug binding pocket on a protein and figure out the shape that needs to fit into that pocket. On the other hand, cryo-EM doesn't require a protein crystal, which is one of the bottlenecks in the crystallography technique. And so it's super useful. And there's been cases where cryo-EM solves a structure that crystallography can't, and vice versa. So they both give really, really important information that help us get structural information about proteins.
Great, thank you. So the next question is for Tom Kirchstetter. Has any of the research shown how long the virus lives on surfaces, such as fabrics?
Well, the research that we're doing in our project in particular, the answer is no. But there's plenty of research about the survivability of viruses on surfaces. In fact, so the research I presented was actually just one part of a larger project, one of these NVBL projects. And that project was called Viral Freight and Transport. And there are scientists at several different laboratories that are actually studying the survivability on surfaces explicitly for part of their research. So then the short answer is yes, it's just that it wasn't part of the work that we were doing in our project at the Flex Lab.
Yeah, I had a little bit of a role in this with the corporation. I mean, I'm sure there's a lot of research on it. I mean, just to give an order of magnitude, which is maybe what the panel was looking for, something like a few hours on copper out to, 3-4 days on like a plastic, and in between for other surfaces. But again, this kind of like particle on a surface transmission is, kind of one of the least probable ways of getting COVID. But.
Got it. Thanks for adding on there. Nigel, let's ask you this question. So do we know why viruses mutate and how the mutations might affect the efficacy of current vaccines?
Yeah, yeah. So, the virus has RNA, which is basically its code, its genetic code that, encodes the different proteins and structure of the virus and functions of the virus. And when the virus is making, many copies of itself, it makes errors doing that. It's not a foolproof system. And so these errors, often they don't lead to any changes. Sometimes, they result in viruses that essentially can't be transmitted. And other times they can relate in changes that lead to changes in the spike protein, for example. And so really from from when we first started to sequence the virus, way back at the end of 2019 and early 2020, we started to see genetic changes in the virus. And as it's become more widespread, we've seen a number of these different changes sort of become more dominant in the populations of the virus that we've seen around the world. There are still, there are many studies out there at the moment investigating whether, some of these changes do actually lead to increased transmissibility, that, do lead to the vaccines not being, as effective. But these studies are really ongoing at the moment. There's a lot of lab studies that have been done, but really, looking into the population, we still have some way to go before we better understand the impacts of these changes.
Thank you. This question is for Thomas. Accelerators are important tools for scientific discovery. How has Bella paved the way for new innovations in medical diagnostics and treatment?
Yes, Dan. Lawrence used radio waves essentially to drive the cyclotron, and that was extremely innovative at the time. Now we have lasers, increasingly powerful lasers. So Bella uses these high power lasers to drive the particle acceleration, and that makes it a lot more compact. So what would previously fit only to a big building can now become a tabletop instrument, enabling a whole new class of applications.
Great, thanks, Thomas, for that explanation. So a question for Jeff. At the end of your talk, you noted that a second generation filter that you're developing would incorporate technologies to make it self-cleaning. Can you talk a little bit about the science behind that capability?
Yeah, definitely. Essentially the most reliable premise for masks being able to, decontaminate or immobilize the virus is electrostatic. So right now we're really doing that, as I showed in my talk, with just electrodes. But there are classes of materials for which they can create their own kind of electrostatic potential, and they can be, kind of made into fabric scale and woven literally into fabrics and masks. And that's something we're testing, kind of as I speak. So That's kind of the next gen. So it'd be kind of like self-cleansing or self-decontaminating. You wouldn't need to go through all of the charging station, all that stuff.
Thank you. And this is a question for Amalie. Why is the iterative process or aspect of machine learning so important, and how will Covert Scholar evolve as more papers are included?
Yeah, that's a really good question. So one of the factors that allowed us to be successful with Covert Scholar is by virtue of being at the lab, we had very close contact with the actual scientists working on COVID. And so we had a very tight feedback loop in terms of design. A lot of the tech companies tried to build similar sort of research literature search tools like Google did, Amazon did, Facebook did, and none of them were quite what the research community needed. They're a little bit perpendicular to community needs. Kind of as an example, we were contacted at one point by people who are working in psychology and well-being, who were doing research around lockdowns and shelter in place and what was effective, who said basically that none of the existing tools service their communities well. And so rather than trying to develop more sophisticated features, maybe we focused on building up our corpus in that area and ended up being the only tool that covered them well.
Thanks, Emily, for that answer. You know what? I'm going to ask one more question and then we'll wrap up. And this question, Tom, is for you. Could you briefly say something about measuring carbon dioxide as a proxy for air cleanliness in a COVID-concerned environment?
Yeah, gladly. That's a good question, and it comes up a fair bit. The main thing is that gas dispersion is an approximation, or gas dispersion approximates the dispersion of the smaller aerosols, which well enough that the experiments are informative and insightful. It's also the case that CO2, carbon dioxide gas, is easy to disperse and also pretty easy to measure at many locations in the room. We had 30 carbon dioxide sensors, for example, and we could be very flexible in that regard. But it should also be clear, like we also experimented with particles of different sizes. You know, we generated 2-micron particles and 10-micron particles. So we'll have plenty of results to share that are specific to particles as well.
Great, thanks for the answer. And we are at the hour. That went by really quickly. So I just want to say before we close, thank you so much to all of our panelists this afternoon. And I also want to thank the audience for tuning in and for your interest in Berkeley Lab. If you want to stay up to date on our research, you can visit us at www.lbl.gov. And you can also follow us on social media. Our handle is @Berkeley Lab. And with that, thanks again for tuning in and have a great afternoon.